Well, hello, ladies and gents. Hope you're all doing well and excited to discuss a topic you've been reviewing since like the sixth grade. We're looking at the cell, all the parts of the cell and how they work together, and what they do to create life. What we've done to this point is the first five chapters looking at everything from atoms to macromolecules, and finally, in the second unit of the semester, we put all that stuff together, and we actually create life. So let's step back and think broadly about the cell theory and what that's discussing. The basic idea of cell theory is that anything that's alive is made of cells. And because of this, cells form this basic unit of life. You put together lipids and they make membranes. You have proteins to do work. You have carbs to provide energy. And then you use DNA as instructions for what to build and how to operate it. So you can take all these different big molecules, put them together, and then you have cells. One feature of cells is that they only come from other cells. So you, as an entity, you're made of about 10 trillion cells. Now, those cells all came from one original cell that began an individual existence, and then that cell came from other cells all the way back. So we know that life was created and survived just once, and now all cells in all organisms are related to one another. So that's pretty profound. All right. So one way to sort of envision cells is by thinking about sort of their size. For a long time, people had no conceptualization of what cells were, but cells have profound impact on life because cells are what do the work of life. Now, one instance of being able to see cells here, it's these little streptococcal bacteria. The strep bacteria, they're like little spheres. And in the case of strep mutans, they produce an acid that eats away at the enamel of our teeth. So for all of human history, people have had issues with dental caries or cavities. And what those cavities are caused by are the actions of little cells like bacteria. Now, how do we get an idea of what these structures are and how they work? We've been able to get greater access to understanding through development of microscopes. Through microscopes, we can enlarge and see images of stuff we couldn't normally see. Now, this is stuff that's not on the AP curriculum, but I think it's helpful for providing context for how we see the images we see. How do we identify what we can see? And one common tool is a light microscope. This is the kind of microscope we use in class. What will happen is you pass light from a light source through your specimen, and then you bend that light using objectives or curved glass, and then you're able to see an enlarged image of that structure. So one fun way I tried to do this is I took a specimen or an image, and then I zoomed in as much as I possibly could. So this is the highest magnification on a light microscope. Again, not fantastic resolution. If we back out a little bit, you can see how it's fibrous and it has some color. We back out a little more, and you start to see there are patterns to the color. If we back out even more, it looks like there are lighter and darker regions. There, it's sort of rough to make out, almost looks like stippling. But you can see almost like a nose and mustache, maybe a mouth. If we back out even further, hey, we have Mr. Cook. So that's what you can do with microscopes. You can take images and get progressively finer and finer detail looking at what's going on. I don't think he doesn't know that I put that in the lecture. So that's a light microscope. Light microscopes are nice because you can actually sometimes observe living cells if your sample is like, say, pond water. So again, something you look at in middle school. But one limit of light microscope is how much you can enlarge the image. You can maybe only enlarge like 1,000 times. Now you can get much finer precision using electron microscopes. So with electron microscopes, the downside is it has to be prepared slides so nothing is living, but you can get a good image of very fine detail. So what you'll do is do this like plating of gold over the sample, and then they bounce electrons off of it to get an image off of that. And so this is what's called a scanning electron microscope. the electrons again bounce off the surface of the structure, and it gives you a three-dimensional view. So this is the interior of the oviduct. It used to be called the fallopian tube, and you see the little cilia, and then the shorter structures are microvilli. And what those do is sweep the egg or the ovum along the oviduct during ovulation or following ovulation. Now, the next image is a transmission electron microscope. Here, the electrons actually pass through the structure. And what that does is it allows you to see inside of these microscopic structures. So this is a cross section of the cilia. So here with the scanning electron microscope, you see it in 3D. You see the surface of the cilia. And then you zoom in with a transmission electron microscope, and you can actually see inside the structure. So these sorts of microscopes give us profound capacities to sort of see very fine level detail about biological structures. So it's really, really cool. So when you see images in your book, you see where they or how they get those images. All right. One question people sometimes ask is, how do you know what the different organelles do? Which is a really good question. So this image sort of explains that. What you do is you homogenize the cell, which means you put them in an expensive blender. So you blend up the cells, and then you put them in a centrifuge, which sort of spins like a gravitron at a fair. And what happens is the densest organelles or cellular material settle at the bottom. And then you can take the supernatant, the fluid that floats along the top of the pellet there, put it in a centrifuge, spin it longer at a higher speed, and do that repeatedly. And what you do is you get finer and finer or smaller and smaller organelles from inside a cell. And then once you have those samples, you can do like functional tests. You can do metabolic tests to see what sort of chemical reactions do or don't happen. And so you can sort of put together the puzzle and figure out, well, if this organelle is present, then these reactions happen. Or if this organelle is absent, then these chemical reactions don't happen. And so then we can begin to tie functions together with specific organelles. So that's how we know what the organelles do. Okay, so the College Board loves to do compare and contrast. One question that's frequently asked is a question sort of tying into what all cells have in common beyond a love of the Golden Girls. Okay, so all cells, any cell that's ever existed, has a plasma membrane. You need a membrane to help control what enters and leaves the cell. So that makes it selectively permeable, where you can sort of concentrate materials inside the cell. All cells have cytosol. That's the fluid part of the inside of the cell. All cells have some sort of genetic material, some sort of chromosome, even if it's just one little circular chromosome. All cells have chromosomes because those are the instructions for making proteins. And again, proteins do the work in cells. And then all cells have ribosomes because ribosomes are the little factories that assemble the polypeptides that will become the proteins. So you need a membrane, you need instructions, you need machines, and you need a fluid medium through which it moves. So that is the basic feature of life for cells. Now, again, they love comparing and contrasting on the AP exam. So it's good to know the differences between prokaryotes and eukaryotes. We talked about similarities, right? Now let's look at differences. Prokaryotes. Pro means before. In carry, well, it means nut or walnut. Apparently, the people that first looked at cells thought the nucleus looked like a nut or a walnut. So anyway, prokaryotes were around before there was a nucleus. They were the first organisms. So they are the more primitive cells. Again, they don't have a nucleus. They have what's called a nucleoid region, which is what you see in blue there. That's the one coiled up chromosome. They don't have any membrane-bound organelles. So no mitochondria, no rough or smooth endoplasmic reticulum or Golgi or anything like that. They're basically a cell with some instructions and some ribosomes. Okay. Bacteria are single-celled, although they can communicate with one another and work in community, which we'll talk about in the next unit. And bacteria have cell walls. And the idea of having cell walls is a structural difference that sort of provides us opportunity to make different types of antibiotics. We can make antibiotics that basically punch holes in those cell walls and make it so that we can blow up some bacteria and kill them. So that's one of the features of antibiotics. Now let's contrast this with eukaryotic cells. Eu means good or true. We have a true nucleus. And having a nucleus allows us to do some really funky things that we'll get into like in January or February. But anyway, eukaryotes evolved from prokaryotes. They do have a nucleus that has genetic information that can be modified in special ways. And then we have membrane-bound organelles. Now, what's the value of this? What's good about having organelles? Well, organelles are basically little membranous sacs. And inside of those little sacs, you can have different enzymes. And if you have different enzymes, they can do different jobs. And if you can do different jobs, you can carry out all sorts of specialized functions. So you have hundreds of different types of specialized cells in your body. And it's because those different cells have different instructions switched on, make different proteins, and can carry out different functions. Okay. Eukaryotic cells, they may be unicellular like protists, like the little things you see swimming around in pond water, or you could be multicellular like us. So we do have a multicellular option in the eukaryotic model. Okay, here's a fair question. Why the heck are cells so small? I don't know. It seems like a reasonable question. And again, it's a concept they cover pretty frequently on the AP exam, so it's a good one to review. But hopefully the work you did where you calculated surface area to volume ratio helps give you some mathematical justification for this. But basically what happens is as cells get bigger, volume increases faster than surface area. Volume is cubed, but surface area is squared, right? So surface area is in the numerator, volume's in the denominator. The denominator gets bigger and bigger, faster and faster. So what happens is as cells get bigger, surface area to volume ratio goes down. Please commit this to memory because they ask questions related to this all the time on the AP exam. Cells get bigger, surface area to volume ratio goes down. So what that means basically is that smaller cells are more efficient. So there's a nice image that you can see here. Here it's overall the same size, right? But if it's just one larger square, it has a surface area of 5,400 micrometers squared. But if you subdivide this into smaller cubes, it's about 3 times as great surface area. So smaller cells are just way more efficient. Here's a more concrete way to try and conceptualize this. I think about Indianapolis. Indianapolis, it's roughly a square. So I've used the city flag as sort of a model here. In the center of Indianapolis, you have Monument Circle. And then in the southwest corner of town along Kentucky Avenue, there's an incinerator that burns up our trash. Okay. So imagine you have a dump truck moving from Monument Circle down to the southwest corner here to burn the trash at the incinerator. If you had 2 little dump trucks going at the same speed, which dump truck would take longer to get the trash to the border? Big Indy or Little Indy? Well, if the dump trucks are traveling the same speed, it would take a lot longer in Big Indy, right? Just because there's a larger volume to traverse. So smaller cells are much more efficient because you can get rid of waste and bring in raw materials much more rapidly. There is a smaller volume relative to the surface area, okay? So surface area to volume ratio really matters because if you get too big, it just takes too long to get rid of waste and bring in raw materials. Small cells are where it's at. All right, let's sort of jump into the organelles just a little bit. And actually, let's pause here. This is a good chance to stop and then pause. So we'll do the little organelle conversation later.